Hello students, myself Popere KS, Assistant Professor Botany of Fresh Shikshan Sanstha's RBNB College, Sri Rampur. In this lecture, we are going to study about gene organization. A part of this topic was left that we are going to discuss in this lecture. So first, let us start with the split genes. So this split genes, it can be also called as interrupted genes. Means what? In this sequence, in the sequence of a gene, there are segments, like there will be presence of exon and introns. So interrupted means, so this is a segment of a gene. Here there will be presence of So here introns and exons are present. Now what will happen? These introns, they are flanked by this exon. So the segment of a gene, or we can say why it is called a split gene, because here there are introns, then there is exon, then again this is intron, and in between exon is there. Now so what is the difference between this intron and exon? What this exon will do, it will this exon, the segment of a gene, it is responsible for the synthesis of a polynucleotide. Or we can say protein. So this is called as, or we can say this is the coding region, which will code for the protein or the polynucleotide, that is a polypeptide chain. But this intron, these are the non-coding sequence, means they will not take part in any translation process or the synthesis of polynucleotide or synthesis of protein. So these are the split genes, which is consisting of intron as well as exon. Now in humans there is 94% of genome, it is 90% of the human gene, they are interrupted, means they have presence of exon and introns. But in case of that is higher organism, or we can say in case of eukaryotes, in yeast, there will be a single interrupted sequence, or there will be very less amount of we can say minority of genes, they are interrupted. And but in case of higher organism, that is in plants and animal, in higher plants and animal, there are most of the sequence of a gene, they are interrupted, or most of the region of the gene is interrupted by the presence of exon and intron. At first it was thought that as intron is not coding for any protein, or it is not passing the information to the for protein synthesis. So it was considered that it is of no use, but later on it was found that even though intron is not contributing any information for protein synthesis, it has an important role for the organism, that is suggesting the structure of a gene, or it have regulatory role for the formation of this protein or other regulatory processes. So intron, it also has some structural or functional role to the gene. So this is about the split gene. Now next we will see the transposable element. So transposable genetic element means, first it was thought that the DNA or the genetic material, it does not move from one place to another. So DNA or the we can say the structure of a DNA, it is stable, or it does not move from one place to another within the chromosome, but and it occupy a stable position, or we can say fixed site on the chromosome. But later on research were done, and it was found that there are elements of the DNA that are mobile, or there are sequence of the DNA which can move within the chromosome. So these are called as mobile element or transposable element. Transposable element, also these are called as jumping genes. And also they are also called as insertion sequence. So these are the name, that is, and one more is there, that is mobile element. So these are the sequence of DNA which move from one place to another within the chromosome, and as they move from one place to another, what this element will do, they will insert at a different site. So it is also called as insertion sequence, and after the insertion there will be some alternation in the chromosome, the sequence of the chromosome. So it will lead to deletion, inversion, or chromosomal fusions and rearrangement. So because of this insertion of the sequence at a different place, this will result in the deletion or other recombination or rearrangement will occur. Now these are also these transposable element. These were discovered by Barbara McClintock. So the scientist, that is Barbara McClintock, he had discovered the mobile or the transposable element in 1965, and he has found the transposable element in maize, which makes the DNA unstable or instability, that is genetic instability. It causes genetic instability in the maize, and why this instability occur? This is due to the presence of transposable element in the maize, and this instability occurs at the site where transposable elements are located. This movement of element is also called as transposition. So this movement of this that is transposable element or jumping gene or insertion sequence or mobile element, this is known as transposition. So this is about the transposable genetic element. Now next we will see the units of gene. So first one is cistron. It can also be used as, or instead of cistron we also sometimes use gene. Or cistron, it is called as unit of function of gene. So it is the unit of function of gene. And what are the functions of a gene now? That is the transfer of genetic information then that is the next is the synthesis of mRNA. And one more is there, that is synthesis of polynucleotide. So these are the functions of a gene, or we can say cistron. Now cistron, it is a unit of function of a gene, and what it will do, it will transfer the genetic information from one to other, that is from the DNA to mRNA or from mRNA to protein. That is synthesis of mRNA and synthesis of the polynucleotide. It is the smallest unit which is functioning as a transmitter of genetic information. Now the next one is, that is muton. Muton, it is the unit of mutation of gene. So we can call it as the smallest unit of a gene on which mutation can occur. And what is mutation? Mutation is the abnormal change in the gene. Suppose this is the nucleotide which will pair with T and G will pair with C. But suppose this pairing occur, that is A will pair with C and G with T. So this is the abnormal pairing. Now what will happen? There will be, so this will result in mutation. So this causes mutation in the gene. So muton, what is muton? It is the smallest unit of a gene in which mutation can occur, and due to mutation, it is a cause of abnormal pairing, which will result in the abnormal functioning of a cell. Now next is the Recon. It is the smallest unit of a gene in which recombination can occur. Unit of recombination of a gene. So in this means it is the smallest part of a gene where recombination occur. And how this recombination means what? So these are the two chromosomes. When crossing over occurs in the phagetin stage, crossing over occurs in the phagetin stage of the meiosis, then what will happen? There will be recombination process will occur. So this is the region, that is the which is forming, crossing over means it is forming the X. So this is X, that is X, it is the crossing over, and it occurs during the phagetin stage of the cell division, and it will result in recombination of a gene. So the Recon is the smallest unit of a gene where recombination can occur, and there may be one or more than one Recon in a cistron. So this is about the gene organization and the unit of a genes. Thank you.